So I was told to talk about my research, but I decided instead to take this opportunity to tell you guys more about the imaging facility and tell you a little bit more about what we're doing on campus as far as neuroimaging is concerned. So I'm really using my role as the director of the imaging facility today as opposed to an independent researcher. Right. So the imaging facility is housed in the psychology building, and we have 2 labs. We have an MR lab, as was stated, that houses a Siemens 3 Tesla whole body MRI scanner. And we also have an EEG lab that has one of very few dual EEG systems, so that we can collect data from 2 individuals simultaneously, and that data can be yoked so that we can do things like look at parent-child interactions or any other sort of social interactions with the system. So I'm just going to tell you in less than 5 minutes now some of the things that we do. So magnetic resonance imaging, we do lots of different types of imaging with MR. The one that we do a lot of in a psychology department is functional MRI. So looking at what parts of the brain are active when you're performing different tasks, right? But we also, of course, do lots of high resolution scanning. So the typical things that you think about an MRI doing in a hospital, we can do. Again, we do have a whole body scanner, so we can image any part of the body. We are a psychological and brain science department, so we only really care about the brain, and that's really all that we do. Another type of imaging that we do spend a lot of time now doing is diffusion imaging. And the nice thing about diffusion imaging is that we're looking at the diffusion of water molecules, and it allows us to look at white matter. So it allows us to look at the structural connectivity of the brain without having to sacrifice a human to do it, right? So we're able to create these beautiful, many of us in the field call the spaghetti picture, right? So it looks like these fiber tracks that are going all over. The interesting thing about diffusion imaging, it's becoming incredibly popular now because a lot of disorders are now being referred to as disconnection syndromes. So many disorders like schizophrenia, dyslexia, autism, where you don't see lots of white matter, gray matter differences, you do see quite a bit of white matter differences. So it's almost as though different parts of the brain are disconnected or not. Information isn't flowing appropriately, and you can get information about those connections with diffusion imaging. And we can also do spectroscopy, again, without having to sacrifice the human to look at concentrations of different molecules. We can do this in living humans performing tasks even. So we have a new study that we're developing now where we're looking at changes in the concentration of glutamate and GABA in vivo in individuals with substance abuse. We're going to focus on alcoholic individuals first. There's a lot of research in non-humans and rats that show that there are these changes in these molecules, but we don't know what's going on in humans, and so we're able to do that using MR spectroscopy. Okay. Skip one. So in thinking about data visualization, so a lot of what because I'm talking to some people from informatics, and we have this new network science initiative on campus. We, over in psychology, are doing a lot of connectivity analysis. So our field has moved away from looking at individual brain regions and focusing on just one node within a network to moving to looking at entire neural networks. And this is both functional networks and structural networks. And so many of you may know Olaf Sporns, who's in the department. He's primarily focused on structural networks, and we're kind of pulling him to say you need to look at functional networks as well. And so he's moved into that direction a lot. And so we do, because we know that no cognitive function, no behavioral task happens with just one brain region. It's the interaction between lots of regions. So we've been borrowing some of the methods and network science and graph theory methods to look at brain connectivity and neural networks. And we've been looking at different populations to see how these networks are altered in schizophrenics, for example. So Bill Hetrick, who's our department chair, and Brian O'Donnell have a long-running study now where we've been scanning schizophrenic patients and looking at these functional primarily, but also some structural network differences between these two groups. We're now looking at autistics and looking at these network differences there in heavy cannabis users. And so many of you may have seen Brian O'Donnell's new grant where we're going to be scanning quite a bit of heavy cannabis users. We've done this in the past, and what I joke with students, because they always ask, how are you going to get people to admit that they smoke marijuana? It's like, that's easy. In Bloomington. What we found in the past that's hard is to find people who never smoked marijuana. Finding those who smoke is not a problem. Finding those who's never ever smoked, the super controls are much more difficult to find. But this is definitely a direction that the field is moving, has moved into looking at connectivity and network analyses. And then to EEG, you'll be able to tell that I'm more of an MR person and not an EEG person by this talk. So I have lots of pretty pictures of MR and only one slide of EEG. That's because it's not really what I do. I'm beginning to learn to use EEG because I'm the director and I feel I should know more about it. So electroencephalography has been around forever, and you see it in hospitals where people, the way I explain it to students is you watch a medical television show and they say, oh, the patient's brain dead. How do they know? There's no electrical activity as measured by EEG. But you can also look at cognitive function with these because, again, EEG is measuring electrical activity on the scalp. And so you can create these beautiful, I don't know how to use this thing, evoked related potentials where these different spikes in the signal correspond to particular cognitive functions. And people have sort of done lots of work for years looking at that. But the nice thing is that people are moving away from just using ERP to doing more sophisticated time frequency analysis, from looking at, as you see here, looking at, in this case, a standard time frequency analysis, but they're becoming incredibly more sophisticated in their analysis techniques, looking at frequency coupling, power distributions across the scalp, all sorts of really cool things, which is kind of fun to see. The field kind of changing a little and moving and using the more sophisticated analysis techniques instead of the standard averaging signals. Now doing a little more with this and finding out that there's much more information in these signals than we thought before. And so that's all I had. I tried to get 5 minutes, but 5 minutes is hard. But if you want to learn more about us, we have lots of information on our website. And so go and take a look. And if you want a tour or if you want to just kind of know more about what we're doing, just email me and let me know.